Today we're going to move on to topic 9. This has to do with DNA, RNA, and protein. We can break these big ideas into the structure of genetic material itself, the way DNA replicates, the flow of genetic information from DNA to RNA to protein, and sometimes I talk about genetics of viruses and bacteria. I normally don't spend a lot of time on this. That's largely because it's so variable. For example, viral genomes can be DNA-based, RNA-based, linear, circular, single-stranded, or double-stranded, and this makes for a lot of information to go through. Each type of virus is going to express its genes and replicate slightly differently. However, in light of the fact that we're dealing with COVID-19, I will talk about this a little bit in an extra video that I'll be posting. If you're curious, COVID-19 is a linear RNA-based virus. But for today, we're going to focus on how your DNA works, how it's going to replicate, and how you express genes. So we're going to start by thinking about the structure of the genetic material itself. So remember that DNA and RNA are polymers of nucleotides. That means that they consist of long chains of different monomers. Those monomers, being nucleotides, are then put together to make the polymer itself. Remember that a nucleotide is made up of 3 major parts: a nitrogenous base, a sugar, and a phosphate group. The possible nitrogenous bases that we can use are going to be one of 2 types. Either they will be a pyrimidine, such as thymine and cytosine, or a purine, such as adenine and guanine. Notice that pyrimidines only have 1 circle of carbon, whereas purines have 2. And they're always going to pair pyrimidine to purine. One of the things that we want to keep in mind when we're thinking about how DNA works is the structure of the nucleotide. When we're thinking about carbons, we're going to count them. So there are 5 carbons that are involved. The first carbon, carbon number 1, is going to be located where the base binds to the circle. Then we count carbon 2, carbon 3, carbon 4, and carbon 5. So the phosphate group is always going to be on carbon 5. Now we have to care about this because carbon 5 and carbon 3 are going to be very important for building DNA polymers, and knowing where they are is going to help you understand how replication happens. Each of the strands of DNA itself is a polynucleotide, which is a polymer, and it's going to be bound sugar phosphate, sugar phosphate. You can see in the picture here that the sugar itself is going to alternate with the phosphate. This is going to form a covalent bond that joins the nucleotides together to make the polymer. That means that we can actually give names to different ends of the DNA. So the end of the DNA that ends in a phosphate is going to be called the 5' end, and the end of the DNA that ends in a sugar is going to be called the 3' end. Now DNA itself is made up of multiple polynucleotides, right? Because it's double-stranded. So the way that this works is that there's hydrogen bonds in between the pairs that hold these strands together. So for example, A and T are going to pair together. The hydrogen bonds between A and T will hold the polynucleotides together. C and G will pair together. The hydrogen bonds will hold those together. So this is what actually keeps DNA as a double-stranded helix. It's important to note here that these strands are reversed from each other. So the strand on the left has the 5' end, the phosphate end at the top, and the 3' end at the bottom, whereas the strand on the right has the 5' phosphate end at the bottom and the 3' end at the top. Right? So these things are in the opposite orientation, and they have to be in order to fit together. So the hydrogen bonds in this opposite orientation are going to allow the DNA to form this double helix-based structure. We can illustrate that structure in a variety of ways. So for example, we have a ribbon model on the left, the partial chemical structure I showed you before in the middle, and a computer space generating model on the right. All of these images mean the same thing. There's always going to be a sugar phosphate backbone that's going to alternate, and there's always going to be pairs of nucleotide bases in the middle. The full name of DNA is deoxyribonucleic acid, with nucleic referring to DNA's location in the nuclei of the eukaryotic cell itself. RNA itself, ribonucleic acid, is unlike DNA in that it uses a different sugar. So that sugar is going to be ribose instead of deoxyribose. It also has a different nitrogenous base, that's going to be uracil, and that will replace thymine. Otherwise, their structures are extremely similar. Now, if you're still unfamiliar or uncomfortable with the structure of DNA and how it fits together, there's a lovely video for you inside the PowerPoint presentation that's been posted, or you can go back to our original topic on how nucleotides and polynucleotides work way back in Chapter 3. The discovery of DNA was really contributed to by 4 different people. Maurice Wilkins was the first person to start looking at DNA and how DNA could be structured. Rosalind Franklin determined that the DNA structure had to be helical in nature, and Watson and Crick really took the information that Maurice and Rosalind put together and built the physical structure. In 1962, they were all awarded the Nobel Prize. Well, at least Watson, Crick, and Maurice were. Rosalind would have received the Nobel Prize, except she died from cancer in 1958. And Nobel Prizes, unfortunately, are never awarded posthumously. The important thing here is that the Watson-Crick model gave a new meaning to the words genes and chromosomes and gave us the information that genetic information in a chromosome is encoded in the sequence of DNA. That's because sequence is the major difference between genes. The structure of the DNA itself is not very different. Now we're going to think about how DNA is able to replicate itself. And that's really important because it's necessary for cells to go through division, and that means it's necessary for us to reproduce. Remember that DNA replication occurs during S phase of interphase. And this needs to happen to let each daughter cell have a copy of every chromosome. There are 3 hypotheses about how DNA replication could occur. The first of these is called the semi-conservative model. In this model, the strands of DNA separate and each one is used as a template to synthesize the new strand. In the conservative model, the entire original DNA stays together but is used to create an entirely new molecule of DNA. And then a dispersive model, different parts of the DNA are used to synthesize other parts, creating no original parent copies of DNA. It's now known that DNA replicates through the semi-conservative model. This means that the 2 DNA strands separate. Each strand then becomes a template for the assembly of the complementary strand, and each new DNA helix has 1 strand with old DNA and 1 strand made of new DNA. In order for DNA replication to occur, first an enzyme has to unwind the DNA double helix. This is possible because the helix is only held together by hydrogen bonds between the base pairs. A different enzyme is going to attach complementary DNA nucleotides to each original half, and this will result in 2 identical sister chromatids. So essentially what we're doing is we're using the template of the original DNA and the fact that nucleotide bases are only going to pair with 1 other base to make a perfect copy of the other side. Now we'll watch a video explaining this and then we'll unpack it a little bit.
In principle, copying DNA, a process called DNA replication, is very simple. The 2 complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing 2 DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding 2 identical DNA molecules.
There are 3 enzymes that we're going to need in order to do what we saw in the video. The first one is DNA helicase. This is the enzyme that will unzip the DNA so that other enzymes can have access to the DNA strands. Using the enzyme DNA polymerase, the cell will then synthesize one daughter strand as a continuous piece. DNA polymerase can only add new bases to the 3' end, so this is the end that's going to grow. The other strand is synthesized as a series of short pieces, which are then connected by the enzyme DNA ligase. So helicase opens, polymerase extends, and ligase connects. Remember that the strands of DNA are different from one another, and we can think of them based on what the end consists of. Ends that have phosphates are 5' ends, and ends that do not are 3' ends. The replication of DNA can only occur from the 5' to 3' direction, because DNA polymerase can only add new nucleotides to the 3' end of a growing DNA molecule. This means that there is a direction that the DNA is going to grow in. If we look at the replication fork here, we can see that on the top strand, this DNA polymerase molecule can synthesize DNA in one piece, because as the replication fork opens, it's still going to be moving in the 3' direction. However, on the bottom strand, that's not the case. As the replication fork opens, a new DNA polymerase will have to join in order to synthesize a new strand. We call the strand that synthesizes one piece the leading strand, and the strand that is synthesized as multiple pieces the lagging strand. And these special pieces have a name, Okazaki fragments. In the end, DNA ligase will connect all the Okazaki fragments together to make one piece of DNA.